Should be sharing the screen and recording. Open up my poster, full screen. All right. Sorry for the height of the video. I can't really raise this, but that's okay. Hello, my name is Joshua Dylan Dodge. I'm a graduate student of the Pellegrino Lab here at UTA, and I'm studying a mitochondrial stress response called the mitochondrial unfolded protein response, or UPR for short. Activation of the UPR is associated with increased lifespan in animals, and the UPR pathway is controlled by a master regulator transcription factor called ATFS1. Normally, ATFS1 is imported into the mitochondria and degraded. However, under mitochondrial stress, this import is blocked and ATFS1 gets imported into the nucleus, and there it controls the regulation of genes associated to mitochondrial protection, innate immunity, and longevity. In our lab, we use C. elegans as a model organism to study the UPR with a transcriptional reporter HSP6 GFP. This gives us a direct readout of UPR activity. So in short, green worms have activated UPR, whereas dark worms do not. Now, in my research, I have found that animals lacking a functional methionine synthase, METR1, this greatly lowers the overall abundance of methionine, and this causes an activation of the UPR and an increased lifespan. To understand how this works, I performed a 4-genetic mutagenesis screen looking for mutations that reduced UPR activation in my METR1 worms. I was able to isolate about 40 independent mutants that reduced the methionine restriction-induced UPR activation. And from that screen, I determined that the C. elegans ortholog of mammalian protein kinase C, PKC1, this is required for methionine restriction-induced UPR activation. Next, I tested some known mitochondrial stressors that induce the UPR, and I found that PKC1 was only required for methionine restriction-induced UPR activation. Now, PKC1 is only expressed in neurons, and it's known to regulate the release of neuropeptides. So to test if the release of neuropeptides is required for methionine restriction-induced UPR, I generated a METR1 UNC31 double mutant, and I found that they had significantly reduced UPR activation. This suggests that there may be some neuropeptides that directly regulate a cell non-autonomous UPR activation. To test if methionine restriction in neurons alone is sufficient to activate the UPR, I generated a string with METR1 only knocked out in the neurons. I found that neuronal METR1 mutants had significantly activated UPR and a significantly increased lifespan relative to the wild type. So our model here illustrates that restriction of methionine in the neurons causes a cell non-autonomous activation of the UPR through PKC1 and the release of neuropeptides. And ultimately, this leads to an increased lifespan phenotype for the organism. I want to thank you all for listening and have a lovely evening.